Widal test. So in this lecture we will understand what is Widal test. It is a procedure in which we will try to find out if the patient has enteric fever or not. Who is causing the enteric fever? A bacteria. What is the name of the bacteria? Salmonella typhi. So is that only the bacteria which is causing enteric fever? No. There are other the brothers of this bacteria like Salmonella paratyphi, Salmonella paratyphi A, Salmonella paratyphi B, there is also C and all. But we will be talking about Salmonella typhi and his two brothers Salmonella paratyphi A and Salmonella paratyphi B. Okay. So what essentially happens in Widal test, I'll just tell you. So let's suppose a patient has come to you, okay, with some fever and you are suspecting he has enteric fever. That means Salmonella typhi infection. Now, if the patient like he already has the fever and everything, the symptoms, and it's been like 1 to 2 weeks, okay, he has all the symptoms. So within this 1 to 2 weeks, he might have developed antibodies against whom? Against the dinosaur? No, against the Salmonella typhi or the Salmonella paratyphi, whichever the organism has infected him. Okay, so his body has developed antibodies against those bacteria. Now, what do we have? Nothing. What does he have? Antibodies. So at this moment the patient has the antibodies, and we also have something. We have the antigens. What antigens? The ready-made antigens. Okay, just like how we go to the shop, cloth shop, we have ready-made cloths, shirt and everything. We have ready-made antigens. So what will we do with this ready-made antigens, and what will we do with those antibodies of the patient? What will we do is we will bring them together and see if there is any reaction. What is that reaction? That is agglutination reaction. Okay, so essentially the Widal test has two total tests. Okay, one is the confirmatory test or the diagnostic test or the screening test, in which we will be trying to see whether he has the enteric fever, whether he has the antigens against, whether he has the antibodies against the salmonella or not. If in the screening test only we find out ki okay, he does not have Salmonella type infection, then why will we do the second step? Not required to do the second step. The second step is the serial dilution one, in which we will try to figure out ki how much amount of antigens is present or antibodies is present in the patient. Okay, so that just tells us ki how much he is suffering from. Okay, so in the first we will just be trying to understand ki whether the patient has the antibodies against the salmonella, or if the patient has the enteric fever or not. So in this, that is a rapid slide test. What do we have? So we'll get a kit, okay, a small kit. Inside that kit we have certain things. What are the certain things? One is we have all this ready-made antigens. Okay, see, there is this O antigen, H antigen. Okay, what is O antigen? O antigen is nothing but the somatic antigen. What is somatic antigen? Somatic antigen is present on the body of the Salmonella typhi. What is H antigen? H antigen is present on the flagella, the tail of that Salmonella. So Salmonella has a body, it has a tail. The body has the O antigen, tail has the H antigen. Okay. What is AH? This is present in the paratyphi. What is BH? That is paratyphi B. So we have two paratyphi, on the paratyphi A, the other one is paratyphi B. So all this, there are this, so total 3 organism I have told you, right? Salmonella typhi. The typhi has O antigen and H antigen, and the paratyphi we are concerned about only the H antigen and all. Okay, so all of these are causing enteric fever, but the paratyphi one causes milder form or less severe form, but the typhoid wala, the Salmonella typhi, it causes severe infection, severe symptoms will be shown. Okay, the patient will suffer more. So these 4 antigens, ready-made antigens, then we have the positive control, the positive one, and then we will also have one more which is not shown here, that is negative control. Okay, and then as you can see here, there is a glass slide which has rings in it. Okay, then you can see a paper here, you can see a glove. What? It's nothing. Let's move. Okay, so this is that slide, glass slide. So what do we have? You can see here we have a ring, two, third ring, four, so 6 rings we have. 1, 2, 3, 4, 5, 6. Oh, okay. What is written here? O, H, positive, AH, BH, negative. Okay, so this is what is written here, so that there is no confusion. Now, what will we do? What do we have? We have nothing. The patient has. No, we also have something, that is antigens. What does patient has? Antibodies. Take out blood from his body. Remove the other parts of the blood and just take the serum. From this serum, what will we do? We'll put the drops of serum on these 4 slides. So we'll put serum here, 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 and what will we put here? After we put the serum here, okay, so wherever the O is written, you put the O antigen there. Wherever the H is written, put the H antigen there. Here AH is written, okay, so we have the AH antigen, you put here the AH antigen, then we have the BH antigen, you put the BH antigen here, okay. And after you put all this, we have 2 more places. Here you put the positive, that one, that white bottle one, okay, this positive control wala you put here, negative control wala you will put here. So, and take this O antigen and put it here and here. So after this, what will you do? After this, just chill out, go to your home, sleep, and then come back. No, you are in the hospital, okay. So after this, what will you do? Take separate, like 6 sticks or something, that thing, what do you call that? So something like that you take and you mix this, okay. After you mix this, what will happen? Something will happen. So what will happen? After you mix, you do something called as rocking. Rock this slide gently for 1 minute and observe the agglutination, okay. So you can just read the procedure here. So what does it mean? It essentially means you have to do the so after you mix all this, you do the ghumar thing, and after that, what, after 2 minutes, what will you do? You will find this thing. What is this? So this is the positive control, this is the negative control. What do you leave all this, you just see this one and this one. See this, what do you see? What do you see? Here you can see small dots. Essentially this is nothing but the clumping, okay. The agglutination has been formed here. Here nothing, smooth, very smooth, okay. So there is no clumping or agglutination here, there is agglutination here. Now this similar kind of thing you can see here also, here also, oh my god, here also. So that means what? That means the patient is positive for O antigen, positive for H antigen, positive for AH antigen, positive for BH antigen. Here's everything, okay. So yeah, that's how it is. So this was the control, and you got the agglutination, the clumping of the antigen and antibody, they both reacted, and you found ki, okay, the patient is positive for the Salmonella typhi. So this was the test to determine whether he is positive or not. Once he is positive, then we will move on to the next one, which was that this serial dilution one, where we will try to identify ki how much he is suffering from, how much load of antibodies he has in the body, or so more load of antibodies means more number of pathogen Salmonella, okay. So O antigen means that he has been infected recently, okay, if this was there. So he has Salmonella typhi also, he has Salmonella paratyphi also, okay. So this was the part 1 of this video, in which we understood ki, okay, he is suffering from the Salmonella typhi infection, that is enteric fever, that is typhoid. Next we will understand the serial dilution one, that will be in next video, okay. Chalo.